Welcome to our Google Hangout on Air with concurrent LCSM chat on Twitter. Today we'll talk for an hour about the changing landscape of lung cancer research and treatment. This Hangout is a joint effort between the National Cancer Institute, or NCI, and lung cancer social media, also known as #LCSMchat. You can ask questions on Twitter, and I, as the moderator, will feed questions from Twitter to the subject matter experts in the Hangout. If we don't get to your question today, we will endeavor to do so in the future. If you have questions you would like answered by the experts, please tweet them during the Hangout and include the #LCSM. More information about the tweet chat is available on the lcsmchat.com website. I'm Janet Freeman Daly, and I'm honored to be moderating this chat today. I'm a blogger, science geek, and metastatic lung cancer patient and activist. I'm also a co-founder of LCSM Chat. I'm joined today by two experts, lung cancer clinicians and researchers. We have Dr. Shukun Malik, the head of thoracic oncology therapeutics at the National Cancer Institute, and Dr. Roy Herbst, the chief of medical oncology at Yale Cancer Center and the Smilow Cancer Hospital at Yale New Haven. He's also a principal investigator on the Lung Cancer Dream Team. David Tom Cook was going to join us, but unfortunately he was called away to a medical emergency at the hospital. Welcome, doctors.
Thank you, Janet.
Thank you very much.
Would you each like to share a little bit about your background and interests? Dr. Malik, if you want to go first.
Sure. So my name is Shukun Malik, and I head the thoracic oncology therapeutics at the clinical investigations branch at NCI. I am also a clinician and continue to see lung cancer patients in the intramural program at the Cancer Center at the NCI.
And Dr. Herbst?
Yeah, hi, I'm Roy Herbst. I'm the chief of medical oncology here at Yale Smilow Cancer Hospital. I'm a medical oncologist, and I've been working pretty much exclusively in lung cancer clinical care and research for the last 20 plus years. I was the section chief for many years of lung cancer at MD Anderson Cancer Center in Houston. I've been here at Yale for 5 years. We're doing a whole large number of clinical trials for lung cancer, from small molecule oral inhibitors of epidermal growth factor receptor all the way up to the newest treatments in immunotherapy. Right now, my current interests are a very close involvement in the lung MAP trial, which I'd love to talk about. We have a lung score for research here at Yale, and also I'm involved, as you heard, in the lung cancer dream team. And I'm certainly happy to take questions or discuss anything related to lung cancer today.
Okay. So first, let's define some terms. Cancer research includes basic research, translational research, and clinical research. What is the difference between these three types of research? Dr. Malik, do you want to go first?
Sure. So basic research is something that, what we study in the laboratory, where we kind of find out what can drive cancer cells. It could be from the tissue, which will be the cells of the specific cancer, or it could be the animal models that we test. So that will be the laboratory basic science research. We test this, growth, the proteins that are involved with it. And then translational is how you take that basic science research into clinic, and also development of the biomarker. So mostly my role at the NCI is the development of the clinical research, which we develop nationally and internationally in cooperation with the multiple groups which are throughout the United States and some international as well. And we coordinate the cancer research through the groups. We have what we call is National Cancer Trials Network, or NCTN, and we have more than 2,000 sites that participate into the clinical research. And Dr. Herbst is involved with the basic science through SCORE, so maybe he can tell us a little bit more about their efforts in the SCORE group.
Thanks, Shukun. I would agree with that, of course. I would say to tackle a disease like lung cancer, which affects more than 200,000 new patients a year in the United States. We need all hands on deck. We have to understand what drives it, the basic biology of the disease, and we have to work in the clinic as the patients come in and treat their cancer. So really we have to go from the lab all the way to the clinic. And in between, there's a large gap. And that gap is translational research. And I would contest that right now the advances are happening quickly from the lab to the clinic because we do have many people working in that space. Take new findings from the lab, from test tubes, from cells that are growing in incubators, understanding things, and very quickly bringing that over to the clinic. And we're seeing, we'll talk today about new advances in this disease. I think there are 3 or 4 new drugs for lung cancer approved in the last year. Incredible. And then we have to take what we're learning in the clinic and say, hey, that's good, but it's not good enough until everyone's benefiting, and then work backwards to the lab. That whole idea that we're working in this space of translation is really what the National Cancer Institute is promoting. Many of us as investigators are working on through a whole bunch of different mechanisms that we're discussing today.
Okay, and Dr. Hertz, you also mentioned the SPORE. Do you want to explain what that is?
Right, so the SPORE is a grant from the National Cancer Institute, and it stands for a Specialized Program of Research Excellence. And it's just that. It's trying to provide funds because, of course, research does take funding, and I would contest that lung cancer is a disease where, given the number of patients, we need as much funding as possible. And these grants are designed in every part of the grant that's going to go from the lab to the clinic, either by developing new therapies that will lead to clinical studies that will benefit patients, or what we call reverse translation, when we take care of patients in the clinic, when we obtain blood and biopsies of tissue, and we work those back to the lab. So here at Yale, working closely with my colleagues, my co-leader is Dr. Li Ping-Shen, very well-known immunologist. We're the group that's working to study lung cancer, both in the clinic, with programs attacking different targets, as well as in the lab, looking at immunotherapy and other molecular drivers.
Okay, and there, if I remember correctly, there are currently 4 lung cancer spores.
There are 4. So there's one here at Yale, there's one at the University of Colorado, there's one in Texas, that's in the Anderson Cancer Center and the University of Texas Southwestern, and then one at the University of Pittsburgh. And we hope to see even more. There are many people who are in official schools that are working together on other grants and projects to work against lung cancer.
Okay. I'm hearing a little buzz in the background. So I can quickly look and see what they're going to do with that. So examples of lung cancer clinical research are precision medicine trials such as LungMap and Alchemist. So Dr. Malik, do you want to tell us something about this? And then Dr. Herbst has some things to say also.
Yeah. So precision medicine is an emerging approach for the disease treatment and prevention that takes into account individual variability in their genes, environment, and the lifestyle of each person. So some of the advances in precision medicine have been made, but it's not currently in most of the diseases. However, one of the diseases that we are really proud of is that we have 2 precision medicine trials ongoing in lung cancer. So there are other precision medicine trials that are ongoing, clinical trials at NCI. I just wanted to mention one is NCI Match, and the second one is the Exceptional Responders. So for lung cancer, we have 2 large trials ongoing. One is Alchemist. Alchemist is a trial in an adjuvant setting, meaning that the patient's tumor has been removed. It's a patient has early stage lung cancer, and now we want to give them therapy that the tumor doesn't come back. So we know that patients who have had lung cancer removed, although all the tumor has been removed, there is a 50% chance that their cancer will come back. And so we are trying to prevent those patients from having the cancer to come back, which is what is called adjuvant treatment. So a standard of care for these patients is chemotherapy. and plus minus radiation depending on if the lymph nodes are involved. Now, what we know from the chemotherapy trials is that we only make a difference or improvement by 4.5 to 5%. So what we in the Alchemist we're trying to do is that we are trying to test the patients for EGFR mutations and also ALK mutations. There are two drugs. One is erlotinib for EGFR mutations and for crizotinib for ALK mutations. What we know about these two drugs or these two mutations is that in metastatic setting, both EGFR mutation patients, when treated with erlotinib, improves their progression-free survival, as well as in ALK-positive lung cancer who have these gene mutations, they will have improvement in progression when they are treated with crizotinib. So in these trials, what we are trying to do is we're trying to add these therapies in addition to standard of care, and we are going to be looking at whether addition of these molecularly targeted therapies will improve survival in these subset of patients. Now, having that said, we also know that so ALK is present in about 6% of lung cancer patients, and EGFR is about 10 to 12%. So in order for us to find enough patients to study these two molecular abnormality targets, we have to screen a large number of patients. So we are screening we will need to screen about 6,000 to 8,000 patients in order to enroll in Alchemist. And what we are also going to do is, since we're going to have such a wealth of knowledge about the epidemiology and the tissue in these patients, we're going to also collect the information on all early stage lung cancer patients. And so that is about Alchemist. And what we have a new arm that we are going to be adding in near future that we're all very excited about is an immunotherapy arm. As we will discuss further, that immunotherapy now has proven to be improving survival in lung cancer patients. So we're going to add that arm also in Alchemist to see if that will also help in early stage lung cancer patients.
Okay, Dr. Herbst, do you have anything to add?
No, except I'll just say that, these trials, these large trials, we call them umbrella trials, because what they do is they take patients with lung cancer, and we know lung cancer is many diseases. Everyone's lung cancer is a little bit different. So we take those patients and we understand their tumors by doing DNA sequencing. So we understand what are the molecular abnormalities within their tumor, and we try to link those abnormalities with a clinical trial. So the idea is we take in one program patients' tumors, we understand them, and then we link that with the clinical trial, and that's a trial that we've been very involved with, known as LungMap. So in this trial, we take patients with one type of lung cancer, it's called squamous cell lung cancer, about 20% of the lung cancers, and we're matching those patients with the right arm of the trial based on their specific personalized profile. And that really is the essence of everything right now. The benefits we're making are because we're doing more personalized medicine, understanding the enemy, understanding the tumor, and then matching patients to the right drug. And through the NCI and through the different academic and community sites and the advocacy groups and the entire community coming together, we built these public-private partnerships where we're using everyone coming together, industry and academia and community and national government, to develop these ways to link the drugs to the patient. Because if we don't do that, that's key. That's critical. We have to bring drugs to patients, test them. If they work, make these approved drugs throughout the country. If they don't work, find another idea.
Okay. Another example of translational work is the lung cancer dream team that you're an investigator on, Dr. Herbst. And that's a great example of collaboration across many institutions. Can you talk a little bit about that and the KRAS mutation work?
Right, so the dream team, you think of the dream team many years back, I guess, with the basketball teams, and when they're bringing together people with different abilities to really go after a target. So that same approach is being approached for lung cancer. These dream teams, of course, from the Stand Up for Cancer, a wonderful group, in partnership with the AACR. And about a year and a half, 2 years ago, there was a call for applications for groups wanting to work on lung cancer. And that was great. It was a $20 million award, so a lot of money. So a group came together, our group at Yale. The two leaders are Jeff Engelman and his group at Mass General Hospital, Fred Walchuk and his group at Memorial Sloan Kettering in New York. And there's Andy Anderson, there's Johns Hopkins, there's a group in Columbia right now, Dana Farber, Sacramento, San Francisco. And this group has come together, and the main focus, the premise of the program, is targeting KRAS in lung cancer. What is KRAS? KRAS is a gene that is critical for tumor growth, and it occurs in about 30% of patients with non-small cell lung cancer. We're targeting that disease through a number of approaches, working as a team. We get together at least twice a year. It's actually, we have calls multiple times a month. And what we're really focused on is how can we target those patients directly, drug approaches, combination drug approaches, with immunotherapy. And working as a team, it's really helping us to figure out how to help these patients. It's good to increase clinical trials, increase research, but what it really does is it brings all groups together. And that really is the critical thing, bringing teams together to target cancer.
Our tech people, I'm getting feedback from the viewers that the volume is low and that people are garbled and hard to hear. I just want to give you that feedback. So we're getting some questions from people through Twitter, and the first one is from Lung Cancer Alliance. Can you tell us about the promise of new immunotherapy agents and what we should expect to see in the future? So, which of you wants to tackle that first?
I can start. Well, one thing that is a phenomenal new advance lately is the fact that we can use the body's own immune system to target a lung cancer. And the way this works is that really in many people, the immune system is already reacting against the cancer. But the cancer is making a protein, and that protein is keeping the immune system from killing it. And we've learned through basic research, talked about that earlier, that we can block that interaction with drugs. And these are called PD-1 or PD-L1 inhibitors. These are intravenous drugs, they're antibodies. What happens is when these are given to patients, about 20% of the time, phenomenal results. Patients who have failed all the other chemotherapies, patients who have the KRAS that I mentioned. These patients, when they get these drugs, the tumor will shrink, and more important, it's a very durable effect that they'll survive for a long period of time. I can tell you I've been doing this now for 20, 25 years. I've seen few examples of such historic breakthroughs in this field. But I am realistic. I see many patients, and I see about 3 or 4 out of 5 don't benefit. So while it's working great, this is the perfect opportunity now for that translational research we talked about a few minutes ago. We have to figure out how do we match the right drugs, the right patient, the right time. That's where we have to develop better predictive markers. That's actually a lot of the work that we're doing here at Yale, at SCORE, and through our group that you just mentioned. That's part of the LungMap trial, develop markers.
Okay, Dr. Herbst, we're getting feedback that you're breaking up and you're kind of garbled on your end, so maybe we can do a little tweaking on your sound. Dr.
Sure.
Dr. Do you have anything to say about immunotherapy?
No, I'm very excited about the arena of immunotherapy. What we do know at this time is that immunotherapy helps, and it does improve survival in second line and further of lung cancer patients. What the future holds is really we do know that certain markers it works best at a level of where the PD-L1 expression is, but we don't have the precise level where it is now, where a lot of research, including Dr. Hurst, is working at this time to figure out which are the patients who are the best helped by this immunotherapy. We do know that it helps even in patients who have a lower level, but who are exactly the patients that it helps. So that is going to be future of immunotherapy. What else is going to be the future of immunotherapy is that it's going to be moving in the frontline therapies. We have a large number of trials that are ongoing in the first-line therapies of lung cancer patients. So we'll find out whether it helps in that group as well. And we have a large number of trials going with the immunotherapy combinations. So this is a field that is very exciting at this time, and the future is going to be very good for immunotherapies, and the precisely who the patients are going to be benefiting, we will know a lot more in the very near future.
All right, thank you. We have another question from online. Are there any trial efforts with a direct goal of increasing the percentage of long-term survivors? I'm not quite sure how to interpret that. I'll let you guys take a cut at it.
I'll start, and that's of course the goal of every trial. And improve survival without having detriment on quality of life. We certainly have to think about at what cost. But what we're really looking for as researchers in this community is for big improvements. We want to find those drugs or combinations of drugs that can really have a home run type effect on patients with lung cancer. And I think the way to do that is using the techniques we're talking about here today. The right drug to the right target at the right time, right patient, precision medicine, using all the tools of biology, of science, of clinical acumen, and that's really what we're doing. And these trials are working. And I can tell you that I just came from conference about an hour ago with our team here, and we see patients benefiting. We also know that we can do even better, and that really makes this the perfect time for research, whether it be NCI, National Cancer Center sponsored research through SPORES, through other grants, through research through organizations like the Dream Team, other philanthropy. We really need to put our resources into this now because we have a lead. We have a very good lead. I sort of look at it like the race to the moon. With that, the ultimate goal being to have everyone have longer survival. We see that we have inroads and we're making great progress. With nivolumab approved, pembrolizumab just approved, 2 immune therapies. Just last Friday, there was a drug approved. I know it is AZD9291. I don't know I can't say yet the name. I just prescribed my first dose of it today. But I'll tell you, we now have drugs that are not the first or second generation, but third generation drugs that target molecular abnormalities. Because we have to stay 1 or 2 steps ahead of the cancer. And that's happening because of all the people that are working in the field and the advocates and patients. I'm sure we have patients on the line now who are really advocating for more research and funding for this disease.
Okay. Dr. Malik, we had talked about a bit about the surgical trials, and we'd hoped Dr. Cook could address them. Can you say something about the CALGB 140-403 trial about limited resection versus lobectomy?
Right, so that trial is still ongoing. We have not finished the accrual yet. Where so standard of care or usually what we have done in surgery is that patients usually has the whole lobe removed with the lymph node dissection. However, there are certain patients who are elderly or have poor lung functions where they are not able to have the whole lobe removed. So this study is looking at those patients whether a sublobar resection is going to be not detrimental in those cases. So that is still ongoing at the time.
Thank you. One of the questions that came in from an oncologist in New Jersey, is there any chance of a matrix array with EGFR, ALK, KRAS, and MET on a chip to speed up the process?
I would say absolutely. In fact, I was just at a meeting in Washington on Tuesday where this was the subject of the discussion, multiplexing these tests. because, again, we have to think about all the information we get is important for patients in real time. These diseases can't wait a month or 2 months. You often don't get enough tissue to do multiple tests. You send a little here and a little here, a little here. So yes, we now have, with next generation sequencing and some of the platforms, I'm quite impressed. We've chosen to implement this here both in research and in our clinical practice, something called the Oncomine technique that's being used in the NCI MASH program. Here you can get 143 genes, and at least in lung cancer, you get almost everything, if not everything you need, one-stop shopping. We've got to continue to do that. Results have to get back to the clinician and patient within a couple of weeks so that they can be used in real time. But that's the goal. And it's also important that we let doctors who might not be oncologists know what type of tissue we need from lung cancer patients, so that at that first diagnosis, the first bronchoscopy, the first CT-guided biopsy, the first obtaining of cells from pleural fluid, we get enough tissue so we can get these results. And it really is impressive that now we have multiple actionable information we can use to help people in real time, which is all of our goal.
And I just wanted to add to that, one of the other research that is ongoing and is going to be very exciting in the near future is the circulating tumor cells or circulating DNA, where for a long time we have had this concern that for lung cancer patients, when they are diagnosed or even after they recur, that we have trouble with biopsying them. So that's another area of very important research ongoing that is in future, that where we can just do a blood draw and we can find patients, tissue tumor that is we can and other things on that.
Okay. Another question that has come in is, are there any combination immunotherapy trials that show promise for patients who have little or no PD-L1 expression?
So I think that I will have Dr. Herbst take this question, because we're going to be opening an arm in Lung Map in very near future about the combination.
Thank you, Shakun. So absolutely, and PD-L1 is a good marker, not a great marker. It's getting better with more and more data coming in. But we do know that there are patients who might not benefit from a PD-1 inhibitor alone. So we already have in our lung master protocol an amendment that should be approved within the next several weeks, which is actually going to look at patients and compare nivolumab, which is a PD-1 inhibitor, versus a combination of nivolumab, this PD-1 inhibitor, plus ipilimumab, a CTLA-4 inhibitor. If you don't want to worry about the details of immunotherapy, I'll just say that we have two ways now of targeting and reactivating the immune system. And the fact that we have this ready to go in this lung cancer master protocol, again, speaks to the public-private partnership, the NCI, the academic community, the NCTN, the National Clinical Trials Network, and the company. In this case, Bristol Myers Squibb, working very closely with us to join the partnership. And the goal is to bring these drugs to patients. And as the questioner asks, now figure out who benefits and who doesn't and why, and try to make this combination available throughout the United States and Canada, because these clinical trials are open at over 800 sites. And I can tell you, I work at a major academic center. Many people come here, but many people can't. And most of the community, most of the cancer is treated, 90+% is in the community. And this point has been really emphasized, especially by the FDA that's been helping us with these trials. We have to get these drugs out to the patients who need them. So we're doing that through our clinical trials, and I hope, Shakun, that the results in a year or two will then provide data that this can now get approved, and if not, we'll move on to something else.
Absolutely, that's very important. That we have patients participating. That's one thing I would strongly say for that. We want your participation in our clinical trials so that we can accrue very fast, and then we can get results very fast to benefit all.
Well, since you're raising the topic of patients participating in clinical trials, I'm in a clinical trial. One of the reasons I joined was not just for treatment, but because I wanted to help make a difference for other lung cancer patients. So what would you recommend that patients or families can do in talking with their doctors to learn more about clinical trials or newly approved therapies?
Well, this is such an era that where there is so much information available to patients, caregivers, that they can easily do search on this, and this is a very exciting time for clinical research, especially lung cancer. So there is, I think the main thing is that when a patient gets diagnosed with lung cancer, it's very overwhelming. It's overwhelming for the patient. It's overwhelming for the patient's family. Is to just get all the information, get educated, understand what clinical trials mean to you, what burden does it have on you, on your caregivers as well as yourself, and then talk to your doctor. And as a physician, it's our duty to explain these clinical trials to you, and then you make an informed consent. That is what I would advocate for, that make an informed consent. Yes, some of the times, as Janet just mentioned, the clinical trial may or may not benefit you, but in order for us to get answers, it is very important that we are, as physicians, able to explain the clinical research to you, and if you get all the information, that you make an informed consent about participation. Roy, did you want to add to this?
No, I would agree with that, Shakun, and I'll just say I'm struck by the fact that despite our best efforts nationwide, I think the figures are 3 to 5% of patients go on clinical trials. So if we could get more patients on trials, it will help us answer these questions, important questions, quicker. But let's look at it from the patient point of view. I think clinical trials benefit patients, because when you go on a trial and you have a disease like lung cancer, where there might not be any standard therapy for what you have, you're looking to raise the bar. You're looking for something different, something better. So patients who went on these immunotherapy clinical trials 3-4 years ago, some of whom were here at Smilo Cancer Center just the other day when we had our lung cancer awareness dinner, these patients are alive now because they went on those clinical trials, and they got the drug now approved many years in advance. So I think it's very important that we promote clinical trials. How do we do that? Well, through the media, through the Google chat we're doing right now. We want to make sure the doctors have the trials available. That's what the Lung Map, the Alchemist trials do. They make these trials available. And we want the patients to be aware too. Because, in anything, even when I go to the doctor with my family and I work at one of the top medical centers, I have to advocate for our care and ask questions. So I think people, the patient learning and advocates helping the patient, then we have the best chance in that conversation in the room with a doctor and a patient. At least one of them might bring up the chance, what about a clinical trial? What's new? What can we explore? And then we can take advantage of all this research we're talking about. Bring the best therapy to patients in real time. But as you said, Janet, learn for the future too. And I think that's very wonderful that you feel that way, that you want to advance the field, but hopefully, you benefit at the same time as well.
Janet, your story is very compelling to me. How you are participating in the trials, and I think that's very heartwarming that you are able to participate in the trial and you feel that there is a benefit to it.
Yes, I had 2 lines of traditional therapy and progressed within a couple months after each one, and then I found to have a targeted therapy available through a clinical trial. I happen to be ROS1 positive, and from my very first scan, I've been no evidence of disease, and it's 34 months and counting, which I'm pretty happy about.
That's right.
I think it's really important for patients to understand that going on a clinical trial doesn't mean you either get the treatment or you get nothing. At a minimum, you get the best standard of care, and it's usually much better care than you would get in a typical clinic. So there's a lot of value in going on clinical trials, and you don't necessarily think of clinical trials only when you've tried all the other treatments and nothing worked, because there are clinical trials available for first-line treatment to find out, for instance, if we can take targeted therapies first instead of starting on chemo. So I think it's important for patients, especially stage 3 and stage 4, to have a discussion with their doctors about clinical trial at their very first appointment. so they can consider those as options.
That's right.
So we have some more questions coming in. How will genomic testing help bring answers for rare cancers for lungs like mesothelioma?
I'll take a shot at that. So certainly mesothelioma is a tough disease to treat. And that's a disease almost always associated, pretty much always associated with asbestos exposure. And there has been a good deal of profiling done with mesothelioma. It's less common than the traditional lung cancers, so that works a little bit less mature. But I have seen that there are a couple of new targets that are emerging. One thing that's very interesting is that the immunotherapy appears to be active in this disease. And there was a very nice report presented last year at the AACR, American Association of Cancer Research meeting, showing very nice activity in that setting. And I know that several drugs now, immune therapies, are being targeted to this disease. And we're continuing to profile and learn, what are the genetic abnormalities that best correlate with outcome. So there is progress in that area, besides, of course, surgery, which is often used for the earlier stages.
So I want to add to Roy's thought process is that we do recognize that mesothelioma is a rare cancer. So we just had a meeting where we discussed the mesothelioma. It was just a working meeting. And so we are going to be in very near future. One thing I wanted to say is we're going to be doing a clinical trials planning meeting about mesothelioma. We want to see how we can develop trials in mesothelioma. I also wanted to say that in clinical center at NCI, there has been some vaccines and other therapies developed, which is directly attacks a protein called mesothelin, which has been actually developed in NCI, and the trials have started, which are going to look into the mesothelin, targeting the mesothelin. In addition, what I wanted to say is that for rare cancers, so it's a perfect thing for patients who have rare cancers like mesothelioma or other, like neuroendocrine tumors and other cancers, to participate in MATCH. So what is MATCH? MATCH is a clinical trial in which it is not important, what we are not looking at the histology, whether they have lung cancer, bladder cancer, breast cancer, any kind of cancer. But what the patients are going to do is that they have had a standard of care, and then we take their biopsy. We study their, through next-gen sequencing, we study what tissue abnormalities they have or genetic abnormalities they have, and then they go into trials, and we have about 10 to 12 arms open right now, and more arms are going to open. So that will give us an idea of whether this specific genetic mutation and this specific drug works on that. So that's another way of participating in clinical trials for rare tumors, and we're looking forward to that. As this trial has just started, we just within the 2 months we have already actually screened 500 plus patients. So we're very happy with the participation of the patients and the physicians in the trial.
Okay. Another question from the tweet chat. Dr. West has one for you, Dr. Herbst. Given work from Yale suggesting discordance in PD-L1 expression, can we rely on it, I assume PD-L1, for clinical decision making?
Right, that's a very good question. So what he's referring to is we've actually looked at different tissue samples and shown that there's a great deal of heterogeneity, meaning that one area of a biopsy might show one result and another area might show another. So what I would say is that a positive PD-L1 result, and there are some new data that I'll present in a meeting toward the end of the year that I think will show this as well. A positive PD-L1 result does help you to feel more confident that your patient's going to benefit from a PD-1 or a PD-L1 inhibitor. The problem is a negative result doesn't tell you that the patient will not benefit. So, we don't want to withhold the therapy to people who might still benefit. So I think that at least as PD-L1 as a marker is concerned, it will help us as we're treating a patient in the refractory setting, meaning they've already failed some chemotherapy, to know that they may or may not have a better chance of benefiting. But the real importance of that marker will then come as we move to the earlier stages of disease and treat patients before they've had any chemotherapy, or in the adjuvant setting, we talked about surgery after they've had something removed. So I think that might be where the PD-L1 comes into play. I'll also tell you that here at Yale and SMILE and elsewhere, through the SPORE researchers and others, we are finding that it's not just about PD-L1. There are other genes that might be involved, and we can measure this through RNA and DNA. And I would predict 2 years from now, when we're having our chat, we'll be talking about a complicated or a complex of markers that we use together to help us to have the best specificity and sensitivity for this. It's still a work in progress, but it keeps getting better. And the good news is, that we're able to treat people as we learn. And it's moving quite quickly because we are getting tissue on most of our patients, something that didn't happen back in 1997, 98, when we first started using some of the EGFR inhibitors. And I know Dr. West was working in the field at that time with us too.
Well, since you mentioned specificity and sensitivity, and we've also mentioned vaccines, there have been some attempts at lung cancer vaccines and also at liquid biopsies, and there are sometimes concerns about their accuracy. Can you explain in simple terms the difference between sensitivity and specificity and why we care?
Sure. Two very different questions, the vaccines versus the liquid biopsy. Why don't I tackle the liquid biopsy, and maybe Shakun, if you want to do the vaccines. But liquid biopsy is very attractive, that you could look at either circulating tumor cells or circulating free DNA, and that would, of course, be easier on the patient. You wouldn't have to stick a needle into the tissues. Two, some people believe it might be even more representative, because you're looking at things that are flowing through the blood as opposed to a tumor where you know that there could be some variability or heterogeneity. The problem is, as you mentioned, sensitivity and specificity. By sensitivity, we want to know that if someone has a specific genetic abnormality, we pick it up. Because, what if it's under-detected because there isn't enough specimen, there isn't enough DNA, there isn't enough of the material that we need to measure present in the blood, or we can't concentrate it high enough. So one concern we have, for example, with the tests that look at epidermal growth factor receptor, is in the blood, we feel that we miss it more than we miss it when we test it in the tissue. So while it might be easier to do it in the blood, we worry that a negative result doesn't mean that that patient doesn't have it. The good news is the specificity is really high. What specificity means is that if you have a positive result, is it really a true positive result? And the one thing that we're seeing is many of these liquid biopsies are quite specific, meaning if you have it, you're pretty sure you do have it, and you can treat based on that. So what really needs to be worked on, in my opinion, is the sensitivity, being able to pick it up more times. But again, techniques have changed. You think about DNA sequencing now, and some of these techniques we use. When I was a graduate student in 1984, 85, I used to sequence 100 bases of DNA, and it would take me a week. And now people can sequence millions and billions of base pairs of DNA, in a day or less. So these techniques are getting more sensitive and better. So I think that we will get to the point where liquid biopsies will become commonplace. They're just not completely ready for prime time yet.
Yes, and I agree with that. So we're not ready for prime time yet, but it's coming. So we have a number of trials that we are going to not change the patient's care based on what we find on liquid biopsies. One of the trials that Dr. Herbst is I believe co-PI as well, is the S1403, where we are going to be testing these liquid circulating cells, but we're not going to change based on that, but we'll be following them. And so what does it mean when the cells come back? Does it mean that the patient is not developing resistance? We'll be testing all that. So there's a number of trials that are ongoing in this field as well. So as far as the vaccines are concerned, so vaccines have been used previously. We had a MAGE-3 vaccine that the trial came back as negative, but we have had a number of vaccine trials that have not shown a benefit so far. But having that said, Vaccines can be that your own immune system works better. So now we have immunotherapies. We also mentioned that there is a vaccine that NCI is working on, which is actually a Listeria bacteria that is injected in patients, which is harmless, but it actually acts and attacks the mesothelin for mesothelioma. So there are trials ongoing, but we'll have to see how the future, what the results come back as.
We did get a specific question about vaccines. Lars in Oslo, Norway asked, what are the thoughts on personalized peptide vaccines?
I would say that I like that idea. I think, the problem with vaccines are, again, they're going to need to be personalized, and it's going to depend on, again, like anything else, what is the tumor making? What antigens are the tumors expressing? And that's where science is going to help us figure that out. And then to make vaccines could help, and that's where a lot of the work that's happening now, using biopsies and studying them in the laboratory, whether it be through the NCI mechanisms, through other grant-funded mechanisms, Center for Cancer, is so, so critical. And I would also point out the caveat that even if you use a vaccine, the immune system might still be inhibited by some of the checkpoints that we're studying and that we're treating with the PD-1s and the other immune agents. So it might be that we need combinations of vaccines plus other things. But I wouldn't give up on vaccines, and I think that someone asked earlier about combination immunotherapy. I personally believe vaccines are going to be one of the big parts of that.
Yes.
All right, from Pidara, MD on Twitter, any progress in the 40% of lung cancers that have no proven molecular targets, I'm sorry.
Shakun, do you want to go first, or should I?
Well, I think that we don't know the targets at the moment, but as the field moves forward, we may know targets. We are also, in addition to, basically immunotherapy, we look at the PD-L1 expressions, but as Roy just mentioned, that even patients who have lower expression, that they also respond. So they don't have a genetic abnormality per se, these patients, but they do have this expression where they work. So yes, I think the other thing is that in the combinations, when we look at the immunotherapy combinations, so far what we know is that they work irrespective of PD-L1 expression in melanoma. So so whether they work in lung cancer as well is to be seen. That's what is the arm in the squamous cell lung cancer in the precision medicine trial for LungMap. We also have PARP inhibitors where they work in some specific gene abnormalities, but when you combine them with VEGF inhibitors, they work irrespective of that. So yes, there are other trials that are ongoing with the combinations that we are going to be knowing more about, helping those patients as well, where we don't know it at the moment. Roy, if you want to add to that.
Yeah, I think that's a good answer. I was on a panel this last Tuesday, actually with one of your colleagues, Shakun, and we were asked this very question, and the question was, are we overhyping the benefits of sequencing and molecular testing? And the answer, I would say is yes and no. Yes. I think that we certainly have made phenomenal advances. Look at Janet. She has a 1 in 100 mutation, and she's on a drug and she's doing great. So we are in about 15 to 18% of lung cancers finding something from the genetic testing that can be used in real time to help people. That's extraordinary, especially considering how common this disease is. But the other 80+%, we need something more. And that's why it really is critical that we think about, what genetic information do we need in real time and what's going to be important for future and for the research. I think, there probably will be a few more of these, what we call genetic drivers, these 1% that we can match with a drug. But I'm thinking we have to look a little bit broader now, not just at the mutations in DNA, but for those on the line who want a little bit more detail in this, at the RNA, it's something called the epigenome, where we look at how the DNA, the methylation of DNA, and how it's regulated. So I think all that's going to be important. But one thing that really is nice is as we figure that out, the immunotherapy seems to be working on a somewhat different population that has some of these most actionable mutations. In fact, 20% of people with lung cancer don't smoke. That's good. But about 80% do smoke or have smoked, and that's okay. We're helping them to stop. But in those patients now, these immunotherapies do seem to work. So I think what we need to do is now figure out what is it about those patients who really do well and continue to treat them with the immunotherapy agents as one single agent. And for those who we can maybe predict in the future by genetic tests, by tissue tests, who are most likely not going to do well, apropos to the question we had a little earlier, those patients might need new combinations. So it really is important for the entire community, the researchers, the funding agencies, and the patients, because the patients have to advocate for this, they have to ask about clinical trials, they have to help us to get these clinical trials through our centers, through the regulatory bodies. I think now, through some of the mechanisms we're talking about, we're making progress even faster than ever before. This new drug that was just approved for people who have refractory lung cancer after they've been treated with an epidermal growth factor receptor inhibitor went from its first clinical trial to approval in in about 2.5 years. Progress is being made and patients are being helped, and I think that's the message I really want to get out during this Lung Cancer Awareness Month.
I just wanted to add to Roy's thought process that the new arms that we're adding to Alchemist, which is an immunotherapy arm, so the patients, all comers can be enrolled in that arm, which is an immunotherapy arm. Similarly, what we're adding to a combination of immunotherapy and CTLA-4 for the lung map, and that all comers can come into those arms. So we are, these immunotherapies seem to work irrespective of the genetic abnormalities.
Right, well, we've had several questions come in related to clinical trials. Patients are wondering, what can you do in order to get over the geographic hurdles and make them more accessible? You mentioned a little bit about that in terms of making trials available in more community settings, but that still is difficult for people who are in, say, rural areas and may not have access. Along the same lines, there was questions of how can we cut through the red tape and increase the access to trials so patients don't have to work so hard to find them. One patient is concerned about the resources he expended trying to figure out if he qualified for a trial, only to find out he didn't qualify for any of them. What are your ideas about how we can make it easier for patients to get into clinical trials?
Roy, you want to take this, or I can start?
Why don't you start? I know the NCI has many good programs for this.
So we are really, I think that really the answer to the access to the patients is going to be through the community-based clinical trials. We do have the sites that are community-based, and we are increasing every few years. We do have the sites that participate in and they are all community-based. So it is a matter of having access to the patients, and I do understand that there may be some hurdles like patients living in very rural areas. So it has been NCI is actively working toward helping those areas to open the sites where they can be participate in the clinical trials. The other thing is that as we advance in the understanding of lung cancer, and it's easier, for example, to have agents available like oral agents and other and things that can be easily taken, that can also help patients to be able to participate in clinical trials. Roy, you think anything else we can work in?
No, I think just make them available, encourage the trials to be at the point of care. The fact that something like LungMap, being a public-private partnership, it provides resources that some people might not have. For example, the molecular testing we're talking about. I don't know the most recent statistics, but I would guess that 30 or 40% of patients still don't get it when it might benefit their care, maybe more. But the fact that you have these trials that can be opened at 800 sites, still not enough. But if you look at the map, hopefully there's something within an hour of many people's houses, at least in the more densely populated areas of the country. Not only do they get the trial, but they get the testing paid for free through this public-private partnership, so that they can have that information both to help them with the trial, but also after the trial is over. And that the physicians are encouraged, and we make it easier for the physicians to open these trials. Things like central IRBs, where the trial is approved centrally and it can get open much quicker. We have to continue to try to get these trials out to the community. And then, of course, the best thing, I guess, Shakun, is the trials accrue quickly where they are open so that the drugs become part of standard of care as quickly as possible. But, it's a constant challenge. I also think we want to get those trials to every different group, and we're using patient navigators, and we have a number of programs here in New Haven. So we're reaching out besides, the main hospital. We have 10 or 12 centers in the Smile Cancer Network, for example, where we're trying to make it easier for patients to get these same trials closer to their home, easier parking, easier for the caregivers who take care of them to come with them.
Okay, we have about 5 minutes left, so I have time for about one question, and then we're going to ask a general question. What important takeaway should patients think about or know if their doctor mentions a trial to them? What questions should they ask? I got you with that one, didn't I?
Well, I'll start then. I think and I get this call all the time, from my patients, but also from friends, family, and others, about clinical trials. I think you have to ask, what phase is it? Is it a phase 1 trial, meaning it's one of the earliest trials, being done, though these days with molecular targets, phase 1 trials actually do usually show some activity. Is it phase 2, meaning it's trying to test a cohort of patients, everyone gets the drug? Or is it a phase 3 trial, which is often a randomized trial? So I think the patient will want to know at what stage is the trial. You'd want to know a little bit about the eligibility for the trial. I would probably ask the doctor or the research nurse or the coordinator or the whoever is helping out in the clinic, what data are available for this drug, what are the potential side effects, and try to evaluate that. You would hope that, again, in this era of precision medicine, that the patient's getting the best trial for their condition at that time. That's what we try to do here. If we know that someone has, for example, a specific molecular abnormality and we don't have that trial open, what I've started to do is, I don't care that they're not going to be treated here. I want them to get the best care. So I try to call my friends in Boston. We're part of the dream team together, or colleagues in New York. So I think it's important that you ask, what is the level of evidence? What is the situation? And is this the best thing? And, if one needs another opinion, there's nothing wrong with getting another opinion sometimes, too.
That's correct. We're always open to second opinions. I just wanted to, this has become little bit more, I wouldn't say complicated, but, we used to say, the patient would come to me and say, okay, it's a phase 1, you're doing dose finding and toxicity. But I just wanted to take home this message on this, that, the earliest patients who benefited from crizotinib were phase 1 patients. So at that time, we didn't know that this drug works in these patients. And actually, the crizotinib, when it was approved accelerated approval, and it was based on the extension of that phase 1. So as we know more of the molecular targets, we find that these, if the drug works, it could benefit even earlier stage patients. So it has become a little bit more challenging that we don't know the answers to everything. But if you benefit, and the patients who benefit from crizotinib were in phase 1.
Well, and another aspect of that is that as drugs are getting approved and then being tried for other cancers, it might be a phase 1 study, but we already know the drug is safe because it's been demonstrated in a previous study. So
Correct.
That's a little different than it used to be. We've got about 2 minutes left, so we want to talk about where people can go for more information about clinical trials, and if we did not get to your question, we will endeavor to get to it in the near future. So suggestions for resources?
There is a clinicaltrials.gov. There is also a 301 number. I can forward that to you, Janet, that you can give patients where they can call NCI number and they can get information about clinical trials as well.
And perhaps you can post the links to that on the LCSM thread in the future.
Yes, we'll do that.
Roy, do you have some?
No, I would agree. I think many of the cancer centers have websites. I would go to the advocacy organizations. I think you can't underestimate how important it is to, and I'm sure maybe you want to share your experience, Janet, but to talk to someone who's had the disease, who's been through this, to give you advice. I know that at many of the meetings that we always have a panel of advocates who talk about where they can find trials and the different resources available, because people should seek these trials out and ask the right questions, and certainly that's going to result in their picking the best option for their care.
Yes, the advocacy organizations do have good resources. I know one in particular, Longevity, has a great site that has lists of questions of what to ask your doctor for different situations. I know the Adario Foundation has a good handbook, and the others have other good resources as well. I would also like to point out that there are online patient communities, Inspire, Smart Patients, several other places where patients have participated in clinical trials, and you can ask them what their experience was like. So I want to thank both of you for participating and for contributing to this. Thank you to the NCI and LCSM chat for enabling us to have this conversation, and I look forward to doing more of these in the future. If you have been watching this, please go to the lcsmchat.com website and take the survey that's at the end of the blog post about this, so that we can see what you liked, what didn't work, and what we might do in the future. Thank you very much.
Thank you.